mapping, QTL mapping, and GWAS. And I hope that after a few hours that I speak about these items, you have a bit more than a basic knowledge about what it is about. So as a starter, I would like to mention the close relationship that is always very important for me, the basic concepts of the genomes. If you talk about genomes, and I take the diploid banana as an example with 22 chromosomes, okay, that is quite easy. You have, you can see 22 chromosomes or 11 chromosome pairs. In the genetical studies, you end up with 11 linkage groups. And at the same time, as you can see in a Nature paper that was published about 6 years ago, you have 11 chromosomes, telomere to telomere, by genome sequencing and assembly. So what I'm not going to talk about much today is about the chromosomes in the haploid cell, the karyotype, but just to mention that we have a general way to explain the parental sets of the chromosomes. The tetraploid line has 4 chromosomes, the hexaploid has 6, and so forth. So tomato, 24 chromosomes, potato is 4x, 48 chromosomes, very related to tomato. But if you take red wheat, it's one of the few examples in which the genomes actually exist on 6 chromosomes. Then there is a genetical and a linkage map. And finally, we also see the physical map or the DNA sequences. And already you can see here on the panel on the right side here that there is an interesting difference between the physical map and the genetical map. Let me see if I can point it here. Yeah, here. So you can see here a clear difference in the length between markers, between a physical map and a genetical map. Oh, now even better in the audience. And this kind of differences became very, very clear, especially around the centromeres, where you have a suppression of recombination, and at the ends where you have most of the genes, and also a very high number of recombination events. So just to make sure that you are familiar with the terms, we talk about the genes as the units of heredity. We can see genes only in a segregated phenotype. If you have a pure inbred, only a pure inbred, it's very difficult to find the genes by genetical mapping, simply because you have no segregation. The place of the genes are the locus or the loci, and these are the position of the genes and the traits on the genetical map. But most of what is now today is very important is the use of molecular markers, which are SNPs or indels, microsatellites, and haplotypes, or haploblocks. I'm going to show that here also. So in the top, you see here the differences between the indels on the basis of single nucleotide, and then below the indels, the insertion deletions, and also the microsatellites, which were very important in the past as one of the markers that were used in the mapping. And finally, nowadays, people use haplotypes and haploblocks to refer to position on the physical map. But now we move on to very much to the basis of what will be the rest of my presentation, the processes of genetical variation. And to make the complete story about what causes the genetical variation, of course we know mutations, but we have to imagine that mutations are rare in terms of how many mutations we find in a single generation. What causes a single nucleotide polymorphism? The SNPs can be spontaneous, can be by radiation, and so forth. The same is by insertion deletions, copy number variations, and sometimes even large structural variations like deletions, big insertions, but also inversions and translocations. Then we have the active transposons. Active transposons, happily enough, They are rare, but when they occur, they can give a tremendous change in the genomes because they can copy large parts of the chromosomes from one place to the other. And happily enough, most of the transposons are most of the time they are inactive. Epigenetical changes, they are common, but most of them, not all, most of them are not heritable. They play a role, but I'm not going to talk about it today. And then finally, which actually is the most important one for today, is the variation that we get by meiotic recombination, which either can be the result of random assortment of homologs or the crossovers between the homologs. And I'm not talking about gene conversions, although I know it is a byproduct of the crossover in the molecular level. Now, just for you to remember, people may say, oh, that's quite boring. But if you come with some critical questions, people always think, oh, that's a bit more difficult than I expected. So here you see the meiotic, the independent assortment of homologs and the metaphase 1. Let me see if we can now. Yeah, now that's better, yeah. So in the independent assortment of homologs, what I mean is that when you have the situation of two chromosome pairs like here, with capital A and capital B and small a and small b, you have two possible recombinations, which actually happen at metaphase 1, so at the end of the first meiotic prophase, in which they can assort randomly. That is the basis, actually, also what Gregor Mendel found more than 150 years ago as a basis of heredity. And the second one is that you have between the homologous chromosomes a crossover which gives a recombination between two of the four chromatids. Okay, if you find 50% recombinance between any two combinations of two genes, and that is an important statement, you can make two conclusions. Either these genes are located on different chromosomes, different so on the non-homologous chromosomes, or these genes are on the same chromosome, but they are far apart, so that at least one crossover takes place. Here, so all the genes between different linkage groups show independent assortment. So all the genes here on this chromosome and all the genes on that chromosome always have 50% crossover. Okay, the second process of meiotic recombination, as I mentioned, is caused by crossovers between two non-sister chromatids of a chromosome pair. I've drawn it here. It's a very basic, a very general statement. I'm sure you have heard this story many, many times. Important is that in a case like here, between locus A and locus B, you have one non-recombinant chromatid, a recombinant chromatid, and a non-recombinant chromatid. Hence, the recombination frequency is 2 out of 4, or 50%. That is the case for two genes that are far apart. If there are no crossovers, probably important to mention, if there are no crossovers between A and B, there's nothing to keep the chromosomes together at meiosis, at metaphase 1. And what happens is that the two chromosomes either get lost or they go to the same pole, and so you get an unbalanced disjoint of homologs. So it is very important. Crossovers are not only the hobby to create recombination, they are one of the stabilizing factors in the course of meiosis. So if a capital A small a and capital A small a are close together, assume that you have here A and you have B next to each other, the crossover probability is far less than 1. You get very few chromatids, hence you can say these two genes, these two loci, they are linked. And 1% crossovers or 1% recombination roughly correspond to 1 centimorgan. And 1 centimorgan is the very basic unit of a genetical distance between two genes. All linkage genes together, they form a linkage group. As you can see in the previous part, here, this is a linkage group and that is a linkage group. And in the end, you will see that the number of linkage groups always correspond to the haploid number of chromosomes. It seems very clear, but later on you will see in the genetical mapping, this information plays a very crucial role. But crossovers is not something like a very stable, easy to understand phenomenon. First, there are differences between male and female maps, and that phenomenon, it is not always the same. This is an old picture from, I think 1977, but I love it, in which De Vicente and Taxley, they make the heterozygous backcross with the tester line for the female, and in the male, they just took the reciprocal, so they used the tester line as the for the backcross, and they used the pollen, the male parent, for the heterozygous. And what you see, to our surprise, is that the female map, the linkage map, is larger always than the male map. So that is what you can see clearly in the female recombination tomato. We're talking about tomato maps. Are 50% longer than male maps. But in Arabidopsis, it is different. In Arabidopsis, you see a picture of a beautiful analysis for chromosome 4 in Arabidopsis, and there you see that the male recombination, the male linkage length, is larger than that of the female. So, in other words, there are gender differences between male and female in terms of how many recombinations you have, but keep in mind that it is not always the same, that female is always bigger or larger than the male. It can be just the opposite. And also keep in mind that the F2, which is the product of a male and a female meiosis, always gives some uncertainties, because you get different, to be honest, you get different linkage map for the male and the females, which is not always revealed by the linkage mapping. There is one more complication that you can see in making a genetic map. Here you see chromosome pair 3 in the electromicrograph. This is the centromere region. And most importantly, if you look at the place where crossovers take place, then you see that around the centromere, a large area of the centromeres, you have no crossovers. Or let me say, hardly any chromosomes. The number of chromosomes in this region is negligible. It means that any locus here and a locus there is tightly linked. You have no recombinant. So they occupy one and the same position on the linkage map. So, in other words, you have to keep in mind that the distribution of crossover events along the chromosome pair is not always the same. Another important thing is that you can count the number of crossover events. You can see the word RN. I forgot to, oh yeah, it's here. It's a recombination nodule. That is the structure that shows precisely in the electron microscope the position of a recombination event, of a crossover event. Now, in this case, in tomato, I can say that on average for this chromosome, you have 2.04 crossovers per chromosome pair. That means in this very case that the expected linkage group is always 50 times 2.04 is 108 centimorgan. Keep that in mind. So many chromosomes have a linkage group length of about 150 centimorgan. And it is never less than 50 centimorgan. Keep that in mind. Probably a good question for later. Now, crossovers can also be influenced by a phenomenon that is interference. Interference is quite tough to understand. It means that if you have one crossover, the second crossovers in general is not very close to it, it's always far apart. And that means that the number of crossovers for a certain chromosome is limited. This is about the median, that is a median. This is about the average. You see many more here and many and less here and there. But if you compare it with the Poisson distribution for crossovers, the Poisson distribution assumed that crossovers are random events, then you see here in the purple distribution a significant difference to what you expected, the Poisson distribution, and what you observed. Now, this phenomenon of the difference between observed and expected can be calculated in what we now call a crossover interference which is comparable to the genetical interference. Now, there are beautiful ways to calculate the amount of interference with a gamma distribution. I keep it I will not talk about in detail, but it is interesting to see here in these linearized chromosomes in the light microscope in which crossover events have been shown by fluorescent tag, that you can see, oh, there's only one crossover here, only one crossover there are two there, and there are two there. It's clear the number of crossovers is limited, and they keep apart. Important information. Okay, now from the geneticist, because the geneticist in general will not look in the microscope to see the number of crossover events. We take the theoretical situation in which we have three loci, A, B, and C, and the estimated recombination between A and B is 0.15, between A and C is 0.2, and between B and C is 0.07. Now, if you think about it, you can see that absolutely the easy way that A, C, B is the best linear order. B is in the middle, and B compared to C gives you the 0.07, and B to A gives you a genetical distance of 0.2. But remarkable enough, if you sum up these two values, you would expect 0.70. You don't see that, you see 0.50. So you see that the estimated recombination on a longer distance deviate from what we expect on the basis of just summing up the small distances. Important take-home message: centimorgan values can be summed up, but recombination values cannot be summed up. For constructing a linkage map, sum the centimorgan values of markers that always very close together. So this is finally is the triangle in which you bring together the position of the A, B, and the C loci. Now, people have recognized this difference between centimorgan and recombination values, and finally, you always want to make the centimorgan values. For that, we need a so-called correction, and a correction actually is correct for genetical interference. It's correct for specific cases of double crossovers in which a mathematical equation is used to convert recombination frequency into centimorgans. We use one of the map functions, Haldane and Kosambi, and to my knowledge, all the mapping programs that I'm going to talk about briefly later always use the Kosambi function. So the Kosambi function is what you see in this curve, that you see that with recombination frequency close to 50%, you see that the map distance can be even 200. And that is what my message is here. Map distances can be added, recombination frequencies cannot. So again, 1 centimorgan corresponds to 1% recombination. 2 centimorgans is about 2% recombination. But 200 centimorgan is definitely not 200% recombination. Why? Because I told you we never can add more than 50% crossovers. In other words, the genetical process of the same parents always slightly different linkage maps. How is that possible? Now, I told you that between A and B in this very important image that we showed you in a while, you have a crossover. But you can imagine the crossovers is on this point, the crossovers is on that point, or there is no crossovers between A and B, and so forth. So the position and the number of crossovers is always arbitrary, they are random. Now, my former colleague, Piet Stam, was a very brilliant quantitative geneticist. He did an outstanding experiment. He used a hypothetical physical map with a large number of markers, and he simulated genetical map just by crossovers that took place somewhere on these chromosomes. And what you now see here, this is a small part of a linkage group obtained by simulation experiments. And you see that the markers are more or less, but not entirely, they are more or less in the same order, in the same sequence, but the position, the length differences, the distance between the neighbor markers is sometimes very, very different. So always keep in mind that if you generate a population of gametes from crossovers between two loci or between a larger number of loci, you always end up with a slightly or a bit more than a slightly different genetical map. That is a very important information. So you see that the linkage map obtained this way is not completely reliable. You need a large number of data. Okay, now finally the crossover interference at the cellular level. This is a beautiful example. Probably some of you have done these experiments as a bachelor student. This is a grasshopper. Grasshoppers here, I've seen many here in Thailand. If you take the male and you remove the testes and make a nice squash or my chromosome preparation, then you see here a bivalent with 1 and 2 chiasmata. Here you see a bivalent 1, 2, 3, 4, 5 chiasmata. You see here bivalent with 2 chiasmata. You see that they all have at least 1 chiasma. That is important. Longer chromosomes have more chiasmata, and they always keep each other on a distance. So if you think about the crossovers, always keep place between 2 to 4 non-sister chromatids that I will mention to you. The number of chiasmata in most species is generally 1 to 4. I'm not talking about some mammals. I'm not talking about butterflies and some other examples where and birds. Birds are very famous. Birds have large chromosomes in the largest chromosome. Sometimes can have 12 or 20 chiasmata. Not in higher plants and definitely not in crop plants. They have what I write here. They have in general 1 to 4 crossover events or chiasmata per bivalent. It means that most plant chromosomes have linkage group measuring 50 to 200 centimorgan. That is a very important statement. If you use a linkage program, and a linkage program on a certain moment after the initial analysis tell you, well, I have a linkage group of 1000 centimorgan, you know that it is simply wrong. It's something you have to remove, you have to correct, and so forth. So the simple first state that a plant breeder can do, if they have a linkage group like you see here, and you have, let's say, a resistant marker like in this position, then if there is a tight linkage of this resistant marker with this marker, and there is also one with the other marker on the other side, you can easily amplify the segment using more markers. And then in the end, you can see, okay, this is my region. When you have a Mendelian marker, when I can map this marker on the chromosome to the sequence analysis, it is that easy. But we know it is often not that easy. Remember that the majority of the traits that we looking at are not Mendelian genetics. Flower color is in general easy. It is a red flower, it's a white flower, or it's a purple flower. These things are very easy Mendelian. But if you look at bigger, larger, heavier, faster, smaller, then you talk about quantitative loci, quantitative traits, and then the story is entirely different. So we're talking about these quantitative traits in a while, but first I will now make the next move. The next step is making the steps towards a linkage map. So here's how you can obtain a gene map data for individuals in a segregated population. I took roughly the example of the AS mapping that will be discussed tomorrow in the workshop, but I just took the tutorial, which for me is very, very clear. It is a tutorial of barley. Barley, like most of the cereals, have a chromosome number of basic chromosome number of 7, and in this diploid barley species, there are 14 chromosomes, and hence it is diploid, and hence you have 7 linkage groups. So here they use 200 plants of a backcross population. Backcross, as I told you, simplify the problem that you have only male or only female meiosis and not the mixture of both, for which 700 markers were obtained. So on average, you have 100 markers for linkage group. The markers on the same chrome but weakly linked may be considered on different chromosomes. But how do you determine on a certain moment if you have two markers and the two markers are 35, 50, 40, 45% recombination? Are they now linked or are they not linked? That is indeed a very difficult question. And that is why I'm not going to introduce another term, is the lot score as a measure for the probability of being linked or not. So I'm not talking about a simple chi-square test and say it is linked or not. It is not that simple. And the lot score, you can say, is a statistical measure used in genetical mapping to evaluate the likelihood, the probability of a particular linkage case. It compares the linkage of observing the data by two loci that are linked or one that are independent. So here in the lot score or the Z value, you have a log 10 value in which you compare the hypothesis in which you assume a given language and you divide by over the hypothesis that you have independent assortment. Independent means that you have 50% crossovers. In practice, if the lot score, so this quotient, if it is more than 3, the likelihood of a linkage for a given pair is 1,000 times greater, because we look at a logarithmic scale, 1,000 times greater than that of no linkage. And this value is, in general, considered as evidence for linkage. And a lot score of less than 2 means no linkage. The lot score, what I mentioned, is a measure for likelihood, it's a measure for probability, and it's very different from a chi-square test. Okay, we come now to the second chapter in my presentation, and that is the use of an F2 segregation population. So the next question is, what material are we going to use for a genetical mapping? I can tell you there are quite a few different populations that can be created. As I mentioned to you, that an F2 is a cross between two inbred lines. You need to have inbred lines. Not always possible, by the way. You get a number of F1s, and the F1s here are identical because you have one red chromosome and you have one blue chromosome. If you make a selfing or you make a cross between two of the same F1 individuals, you get a number of F2 individuals. All these F2 individuals are different, and as you can see here, I'm trying to draw them very clearly. The chromosome, the maternal and the paternal chromosomes have different positions for the crossovers. So you see that all these individuals are different, but they also have recombinations at different sites. Now, what is now the attractive thing about an F2 segregated population? It is simple and it is fast, because you have only one generator from this to that. And the allele frequency is 1 to 1, Mendelian, codominant, or it is 3 to 1 if it is dominant. And this information is essential because quite a few of markers will have what we call segregation distortion. Let's say if the 3 to 1, if the class of 1, the recessive one, as we tend to call it, if the 3 to 1 is close to a sublethal factor, then you find not 3 to 1, but you find 4 to 1 or 5 to 1 or whatever. So in those cases, you have segregation distortion. They occur quite a lot, and we have to keep that in mind, or we have to correct. Because the method is simple and fast, one generation, the method is cheap, but it has a lot of drawbacks. Parents must be inbred. They must be completely homozygous. You can do that often, but there are several, or better can say, there are many crops which do not allow pure inbred because they get too weak. You have to keep in mind that the combination of maternal and paternal recombinations that differ in average crossover frequency. Few recombination events means a low level of precision. Here you have seen 1, 2, 3 crossovers one crossover, no crossover. How can you explain no crossover? Keep in mind that when you have a bivalent with one crossover, two of the chromatids form gametes with zero recombination. So that also can happen. So few recombination events means a low level of precision. And the samples of an F2 population with the same parents are never the same. So you make an F2 population and you give it to a friend in another place and say, please use that also for making a linkage map. You always end up with slightly or a bit more than a slightly different genetical map. And this kind of an F2 segregation is limited in using the phi mapping and QTL mapping. What are the alternatives? Now, one alternative is, I have to say, not used very often, and these are the so-called chromosome substitutions. We did it in my lab about 10 years ago, and the procedure is that you have, from an inbred line, you have F1s, but in these F1s, you block crossovers. You simply knock out essential genes that are involved in crossovers, and the haploids that you obtain from that are used for making a double haploid. A lot of work, I have to say. So, but anyway, you see here that all these chromosome pairs in this double haploid are either blue or either red. So you have, is it interesting? Yeah, this is interesting because later you can see the trans effects of QTLs between different chromosome substitutions. So what is the effect of a QTL? Not in cis, so not on other markers in the same chromosome, but on QTLs in other chromosomes, chromosome substitutions. So it allows the analysis of epistasis to remind you, gene-to-gene interactions between chromosomes, and the reciprocals can be used to create an original F1. We showed that, I think it was 12 years ago, in a Nature Genetics paper, and we got a lot of remarks, especially breeding companies were afraid that the F1s can be reconstituted, can be recreated by the competitor. Now, the truth is it is not that easy, because knocking down meiosis means that you need transgenic approaches, and making a double haploids is another effect that takes quite a lot of time and effort. So what I said, it requires knockouts of a crossover machinery and haploid selection. The alternative is many generations connection, and it is time, it's not one time, time consuming, and it is expensive. Now we come to the most important class of what we call immortal populations. Immortal does not mean that a plant never will die. No, it means that the genetical information of every plant in an immortal population will, by selfing, give an offspring that is completely identical to the mother plant. That is what it means, immortal. It is immortal in a genetic sense. And the first that I mentioned are so-called recombinant inbred lines. How do you obtain the recombinant inbred lines? Just think about 2 slides ago, when you have an F2 family, an F2 population, and every plant is selfed. And what you see after selfing, that the percentage of heterozygosity from 50% goes down to 25%, or 1/4. And why? Because remember, in an F2 you have capital A, capital A, capital A, small a, small a, small a, in the ratio 1:1. And in the next generation here, the capital A, small a, again gives a 1:1 segregation, and it will, every step, it will decrease the percentage of heterozygosity by 50%. 50% here, 25%, 12.5%, 6 point, you can also say half, a quarter, at 1/8, 1/16, 1/32, and so forth. And in the end, after 8 generations, 1, 2, 3, 4, 5, 8, after 8 generations, you have almost only plants that are homozygous, but they retain crossovers of the original plants. Now, keep in mind that in this plant, if you have here, you see my pointer. If you have a crossover here, it has no effect. But if you have a crossover here of this effect, you get a new crossover size. And that creates finally this kind of plants, which are recombinant inbred lines. Is there heterozygosity? Yes, there is still residual heterozygosity of 1.8%. That is what we call negligible. So in summary, the pros are the advantages. Different seed samples, it's the same real population, have identical genotypes and are very suitable for QTL mapping. The same rules in different sites show the effect of the environment. So my very appreciated colleague in Wageningen, Professor Martin Cornet, he was one of the first to produce large numbers of recombinant inbred lines in Arabidopsis. Actually, he did not do it. He had a very appreciated assistant, and she worked for many, many years and produced a large number of recombinant inbred lines between accessions and Arabidopsis. And with that, she could distribute this material for free to other labs in America, in Asia, everywhere in the world. And it gives you a great advantage because you talk about panels of recombinant inbred lines in different labs where people can look for genes, for genetic effects, for environmental effects, whereas you know that the genetical content of this material, of this library, is entirely the same. So the drawbacks of the material is keep in mind that you have 8 selfie generations are required, we said in general, to get a residual heterozygosity, so a final heterozygosity, which we call negligible. And every generation thereafter, the percentage of residual heterozygosity will be cut by half. It is time consuming to make. If a crossover is insufficient, you need not an F2 generation, but you start with an F3 generation, and then you get by itself, of course, many more crossover sites. As a sort of alternative to recombinant inbred line, people thought about using double haploids. Again, keep in mind we start with the F2 population. And from the F2 population, we obtain haploids by in vitro culture, as you can see here, in vitro culture of anthers, or microspores, in some cases even ovules, to produce haploids. And the haploids can be doubled by oryzolene or colchicine, or in Arabidopsis, if you have a haploid plant and you grow it in your growing chamber, you can see some branches which produce spontaneously double haploid, and then you are happy, then you have precisely what you want to have here, an immortal population of doubled haploids, which are perfectly 100% haploid, homozygous. So as the recombinant inbred lines, they work as perfect as well. It is a fast method for a one-step homozygous recombinant genotype population. Fast, perfect, but not all crops can produce in vitro haploids. And if you are really a master in producing haploids for different crops, I can tell you breeding companies are very interested to hire your service, because this is in general an expertise, a skill that only few people have. It's difficult to say how you do it, but some people are more lucky or more skillful to produce haploids than others. And you have only one male meiosis, which gives a limited number of crossovers. How you can solve that? Okay, you can solve that by the number of recombinant genotypes. You can take much larger. So instead of 200 plants for a double haploid population, you can say, I take 400 plants. Or you make a double haploid population of an F2 or F3, so an F3 generation. Then you also have a higher number of recombinants, and so more information for your genetical mapping. So this is a very successful method, but not always applicable. Once you have recombinant inbred lines, or you cross it back with a heterozygous F1, you get this kind of situation in which one chromosome is recombinant and the other is not. You make a couple of back crosses the same way as recombinant inbred lines, and then you get recombinant inbred lines that contain only a very small segment of the blue chromosomes, a small, what we call interbred segment. And that is very good if you want to map for sure your QTLs on the chromosomes. So the advantage is that the strategies can be used for using PC1 and integration lines with chromosome regions from less related genotypes. The good thing is you have no epistasis, because you look only at the QTL in this or that, on that, on that, or that place, which is not influenced in cis or in trans by QTLs from other positions. However, it is time-consuming, labor-intensive, and nils are more powerful than rils, but can only be used to study one locus at a time. Then we get to more advanced, the more modern lines that have been created. One are the so-called multi-parent advanced generation intercross lines. Thank you very much, Nick, for your suggestion. He said, what does magic means? That is the full word. So what you do in that case, you start with 8 different genetically very different parents. You cross 1 with 2, heterozygous, 3 and 4, heterozygous, 5 and 6, heterozygous, and 7 and 8. So you have on a certain 4 very different heterozygous plants. These are crossed again and again. So in the end, you get a multi-parent line. And also that one, we use the same trick of inbreeding a self in a self in a self in to remove all the heterozygosity. So these are rils obtained from multiple parents. It is very time consuming and so it is labor intensive. But that doesn't matter that magic lines are produced for all major crops. I remember they use a lot for tomato and for barley and several others. So they are more informative, but they can only be used to study one locus at a time. And then finally, we have the natural collection of accessions and cultivars. So we make outcrossing and recombination. We make all kinds of different combinations. And in the evolution, we know they shovel around a lot of chromosome segments. And finally, you end up with the current allelic state panels, which have chromosomes that have components of a large number of accessions and cultivars. So you do essentially, you repeat the evolution, but you do it in a couple of generations. These mixed populations, they contain subpopulations of different genetical backgrounds using sequenced and genotyped germplasm collections. Very, very great and informative. But they are expensive to develop and they are labor intensive. The populations need to be tested for possible structure properties, population structure properties. I'm going to talk about it later on, because this point is going to be a very very important issue when you use genome-wide association mapping. What is the idea? In short, if you have this guy here, this is one of the accessions, and think about tomato. Tomato, if you have here a Lycopesicum peruviana, Panella, we know that this wild relative has several inversions and so, and that means it has very profound effect on the panels of natural collections of accessions that you use later for the genome-wide association mapping. And there you see that this kind of structural differences between the original accessions, cultivars, and wild relatives, and also wild relatives, then you get a problem. in the population structure. We'll come to that later this morning. And then finally, there are special mapping populations to keep in mind. One, what is of course important is that you have several crop species in which inbreeding is not possible. Why is it not possible? Why is it not possible? Because inbreeding will make the next generation weaker and weaker and weaker, because you get the accumulation of deleterious mutations. But you can, for the mapping populations, you can take two different heterozygotes and you can make crossings of them. But the trick that we are going to use for normal F2 generations is not possible. So we need a program for outbreeding full families, like you see here. The parents are highly heterozygous, outbreeding genotypes, and you need special mapping software. Now, happily enough, I can tell you, nowadays we have far better algorithms to construct genetical mappings, and with it, nowadays, it is possible to produce for every chromosome a maternal and a paternal linkage group. And then the final thing is that I'm happy not to discuss it today because it's a very complicated story, is the story of mapping tetraploids, mapping populations of autotetraploids. I'm not talking about mapping populations of an allopolyploid, an allotetraploid, because they behave like a diploid, then you don't have the problem. But you have an autotetraploid like potato. Potato is a very well-known example. And then every chromosome has two homologs, and every chromosome set consists of two chromosomes, homologs, and two homologs. So here the two blues are the homologs, or the identical chromosomes, and the dark blue and the light blue are the homologs. See here, capital A, capital small a, or you can say A prime, A prime, and so forth. So every chromosome has two homologs and two homologs. That is difficult. Why is that difficult? Because in meiosis, you can have preferential pairing. And preferential pairing means that for the pairing and crossover events, the plant or the meiosis preferably exchange segments between the homologs or between the homologs. So preferential pairing is one difficult thing. And the second, I'm not going to discuss that, but just mention the name, is what we call double reduction. Double reduction is the result of sister chromatin exchange. And then you get a completely different segregation of markers in your progeny. That makes it very difficult. But don't worry, also for autotetraploids nowadays, we have very powerful software that we can use. Okay, now finally for this part, I just would like to mention the construction of linkage maps. Tomorrow we will talk about ArcGL. I understood that Puimpak told me, but there is an improved version, ArcGL2, an improved version, but the format of the data file is different, so we will not use it this time. And for the linkage mapping, ArcGL can be used, but it's very slow when you have a big data set. And tomorrow we will use ASMap for better algorithms, and we're much faster. And there is another recently published, also in the R libraries, is the package CSUM. The procedures are more automated. I was not able to use that now, but that, and then you have, of course, you have the commercial packages like JoinMap, but they are very expensive. So, in the preparation of our data, Ajahn Puimpak mentioned to me, how good are my data? Well, use the program Flipjack, very powerful. You can visualize the genotypes, and here also you can download Flipjack for free, and then it works on Mac and Windows and so forth. Very, very powerful. And as you can see here in this example, you see which have the A, which have the G in this, which are missing data, which have to be removed. And in this case, you see the situation that the plant is heterozygous. So it is a graphical representation of the genotype values for each chromosome, for each homozygous or heterozygous allele within the current chromosome. The data are shown horizontally, and the data for the markers are shown vertically. By default, FlatCheck will show you one color per allele type. That's a different situation. Not talking about an altered data point. Okay, now the final part of this segment in my presentation about the linkage mapping is about what are the major steps in the ASM mapping, linkage mapping, and that is what you see here. For first is the different steps, showing them how to clean the data for a robust and reliable linkage map. I did not work much with AS mapping. In the past, I worked with join map, and join map essentially goes to the same sort of steps that we use here to clean up the data to finally make a linkage map. Keep always in mind that in the end of your cleaning up, the number of linkage groups should be equal to the haploid number of chromosomes. So you load the data, in this case, that was given in the tutorial. It is in barley, not barley, but barley BC1 with 40 chromosomes, and you check first for the missing genotypes, and the missing genotypes are removed. That can be a quite substantial part of your data. Then you remove clones. What are clones? Clones are the markers which are genetically contain the same genotype. They give you a wrong impression for the program, so you remove all the clones so that you end up only with one informative genotype. And you remember I talked about the segregation distortion is also an aspect that can play a very bad role in trying to make a linkage map. And a segregation distortion means that your alleles for a given marker is not a 1-to-1 or the 3-to-1 ratio, but can be 1-to-2 or 1-to-3 and so forth. And it must be removed, but later, eventually, you can put back these segregation distorted markers. In the example here, then you see on a certain moment, you see a figure like that, and you have a linkage group 1, 2, 3, 4, 5, 6, 7, 8, 9. This is remarkable for two reasons. The first reason is that you can see that this linkage group is very small. And this is also far too small. That one is certainly too small. So you see that some of the linkage group must be merged. Because remember, we have 9 linkage groups, and barley has basic chromosome number of 7. So we have to clean up further. So the greater data set shows 9 linkage groups, whereas 7 are expected. So the genotypes that have recombination rates significantly above the expected recombination of 40 are then removed. So we use the Kosambi function to correct for transferring the recombination frequency into centimorgan, correction for double crossovers. And then still we have a number of linkage groups that are too short. You clean up your data, you end up the segregation distorted markers, and then what you can see here in the end, I go a bit fast, but if you do the tutorial, very worth the trial, then you see here that you have 1, 2, 3, 4, 5, 7 linkage groups. In a very short nutshell, I tell you the story how to make a linkage group. And this linkage group, with the peak where you have a lot of recombination, blah, 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 and so forth, will give you the information how to make a linkage map. I have also to mention, I mentioned already a while ago, that AS mapping is a recent mapping algorithm based on an algorithm that was invented by Dr. Wang, if I remember well, from about 10 years ago. in which the mathematical method to reshuffle your data to find strong linkage is far better than RQTL. So if you for QTL, you probably may need a few days. This program can do it in less than half an hour to make the same linkage mapping. So very, very warm, recommended. Unfortunately, I have no information about CSAP, which is supposed to be more automated, but it doesn't work on my computer.